Now, let's move on to non-specific chemical defenses against pathogens. We're going to be talking about toll-like receptors, or TLRs, NOD proteins, interferons, and complement. First, toll-like receptors and NOD proteins. Toll-like receptors are receptors that are found on the cytoplasmic membrane or in the phagosome membrane of cells, mostly phagocytes. Toll-like receptors bind to what we call PAMPs, or pathogen-associated molecular patterns. These are molecules that are never found in the human body, so they are identified as being foreign. Now, this table shows some of the toll-like receptors and what they bind to. You don't have to memorize these, but get an idea of what a PAMP is and what a toll-like receptor will bind to. Now, some of these you will be familiar with because I've emphasized them through the semester with an eye toward this chapter when we start talking about PAMPs. Now, NOD proteins are not found on membranes. They're found within cells, but they do the same thing that TLRs do. They bind to PAMPs. TLRs and NOD proteins are binding to PAMPs. Then what do? Well, they stimulate the cell, if it's a phagocytic cell, to adhere to the pathogen and phagocytize them. They stimulate cells to release interferons, which we're going to be talking about later. They stimulate cells to release chemotactic factors, which we've already talked about. Stimulate the cells to release chemicals that cause other immune cells to develop. And in some cases, they stimulate the cell to undergo apoptosis, or programmed cell death, or you can think of it as cell suicide. All of these are important in the innate immune system. We've found that when there have been mutations in toll-like receptors and NOD proteins, it leads to disease. Next up, interferons. Interferons, in broad overview, are chemical signals that protect cells that are near virally infected cells. So let's take a look at how this happens. Here's this cell, and it's become infected with, in this case, a virus that has double-stranded RNA. It's too late for that cell, but when it senses that there is a virus inside, the genes for the interferon turn on, and the cell produces the interferon proteins, which it releases, and it is picked up by neighboring cells on their receptors. This stimulates transcription and translation of antiviral proteins. Now, meanwhile, over here, this cell has been devoted to making new viruses, and it ruptures and it dies. Well, when these viruses attach to the cells that have produced these antiviral proteins, which are inactive at this point, when they bind to the genome of the virus, they become active. And what they do is they bind to the ribosomes of the cell, preventing them from attaching to mRNAs. They also go through and snip up all the mRNAs, the cellular mRNAs as well as the viral RNAs. When you take out mRNAs and you prevent ribosomes from attaching to mRNAs, this shuts down protein synthesis within the cell. This can last anywhere from about 2 to 3 to 4 days. Basically, the cell stops producing proteins, including the viruses, and the virus becomes inactivated. There are also RNases that go through and clip up the viral genome, and the cell survives. Now, all of this is very cool. We've been working on using interferons to treat chronic viral infections and also to treat cancer. Now, when you have a chronic viral infection, your body on its own has not been able to clear out these viruses, so we inject large amounts of interferons to kind of give the body a boost. Now, you feel horrible because you're not producing proteins in any of your cells. I've been told that it's like the worst case of flu that you've ever had. You're miserable, muscle aches, you're tired, you don't feel good. Sometimes there's a little bit of fever for about a week, and then you're better. People who have chronic hepatitis B or hepatitis C, we aren't able to isolate the virus from their blood for up to 2 years. Then they need another interferon treatment, but that's not bad. Every 2 years, not bad. This also works against some types of cancer, because when you shut down protein synthesis in cells that have lost their ability to control their cellular division, they die. They're unable to stop and wait out the lack of protein synthesis. So this has been effective too. So interferons are pretty cool. Next up, complement. Complement are serum proteins that have various functions. They act as opsons. Remember opsons? When you have, in this case, complement attached to a pathogen, it enables phagocytes to phagocytize better and faster. They act as chemotactic factors. They attract phagocytes and neutrophils and other innate immunity cells. They can be indirect triggers of inflammation and fever, which we're going to be talking about later. And what I consider to be the most cool thing, they form membrane attack complexes. So now we know what they do, let's talk about how they do it. Complement can be triggered in 3 different ways. The classical pathway, which involves antibodies, the alternative pathway, which involves PAMPs, and the lectin pathways, which involves mannose, which is a carbohydrate which is very rarely seen in animal cells. Now, what happens is these various complement proteins, and there's a ton of them, they start splitting. And then they become activated once they're split, and they go and split other complement proteins, which cause other complement proteins to split, all of which triggers what we call a complement cascade. Now, a complement cascade, its intent is to amplify the signal. So, say you have one cell that's covered with antibodies, you end up getting a whole bunch of complement activated from that one signal. Think of it like a radio, turning up the volume dial. That's what the complement cascade does, is it turns up the volume on the signal. All of which ends up causing opsonization, or increased phagocytosis by phagocytes, stimulates inflammation, and we form membrane attack complexes. All of this is quite a bit. Here's what I want you to remember. I want you to remember the 3 ways that complement can be stimulated: the classical pathway, the alternative pathway, and the lectin pathway. By the way, the classical pathway is part of the adaptive immune system because we have antibodies involved. The alternative pathway and the lectin pathway are both part of the innate immune system. We don't have to have antibodies or any part of the adaptive immune system to cause the complement to cascade. Then, after it's cascaded, we get opsonization, inflammation, membrane attack complexes, and in some cases, fever. Let's use the alternative pathway as an example. So, complement binds to, say, endotoxin or LPS on a gram-negative bacteria. That causes a complement cascade. Now, because there's complement on the outside of this gram-negative cell, it makes it easier for a macrophage to phagocytize the gram-negative cell, because the phagocyte has receptors for complement on its cell membrane. Makes it easier to grab, right? Even if, say, this cell happens to have a capsule, which would normally make it harder for a phagocyte to do its job. With inflammation, because we've got chemotactic factors that are causing phagocytes to enter, that cause macrophages and neutrophils to enter the area, that stimulates inflammation, also stimulates capillaries to dilate so that those cells can enter the tissue. And then in the next slide, we're going to talk about how membrane attack complexes form. Now here, there's a whole bunch of stuff down here that for this class you don't need to memorize. But it is good to take a look at it. So here's a pathogen. We've been exposed to this pathogen before, so antibodies attach to the pathogen. On the FC portions of the antibody, and also on some of the other portions of the antibody, there are sites for complement protein to bind. Once they abound, they start splitting these other complement proteins, which reform into enzymes, which split other complement enzymes, all of which ends up, in this case, forming in a membrane attack complex. We get complement proteins forming this ring in the cell membrane. And if you have a ring or a pore, then fluids and ions and molecules can flow in and out of the cell, causes the cell to burst, causes them to lose their integrity, and they die. Isn't that cool? Macs are so cool. In my opinion, it's the coolest thing that complement does.